Okay, so in this protocol, I'm going to show how to quantify Western blot data using ImageLab. So this is ImageLab 6.1. You can find it both on the lab computers. You can also download it yourself from the Bio-Rad website. So it's a program produced by Bio-Rad. It's freely available. If you go to biorad.com and then log in. You have to set up an account, free account, and then you can search for this software. You can download it. There are instructions in the handbook. Again, it's freely available, so you can download it onto your computer if you'd like. So I'm going to open the files. What we're going to do is open the files that we want to quantify. So, and this is assuming we've done the Western blot and we have a signal we want to quantify. And maybe make sure I open the right one. So I'm going to this is the Western blot I did a few weeks ago, where I was looking at XPA expression in Hackett cell lines where I knocked out, or have some clones of Hackett cells where I've knocked out the XPA protein. And so I'm the right ones. Let's see if that's correct. Let's see, sorry about that. Let's see. So that should open. Okay. Let me pause for just a second. Okay, so I had a problem with one of the files here. One thing to note is that all the files need to be black and white. So on the Azure Imager that we have in the lab, can't take images that are colored, which is fine. The issue is this software cannot open multicolor images. So on the Imager, and somebody could show you, is you can split it into the individual red, green, blue channels and then save it there. And then it's essentially just black and white. So it has to be black and white for the image to open. And so the Ponzo blot was a problem for this. So essentially, I'm going to open 3 files here. So I've got one is the Ponzo stain, which stands for total protein. We can use this to normalize protein expression, because ideally we would have identical amounts of protein lysate in every lane of the gel. Sometimes it's not exact. And so to normalize to that differences in proteins, we can use the Ponzo stain. So what we're going to do is we will quantify the region of the Ponzo blot. We have our blot where we probe for XPA. I'll go more over that in just a second. And we also have a white light image that has the picture of the membrane itself. The advantage of the white light image is it has the molecular weight markers. Again, that is posted by the gel running area where you can see what the exact molecular weight of the individual markers are. So one thing we want to do is to merge the two, and that's useful just to have a picture where we can show, again, this blot shows several different bands. And so that could be a common occurrence of Western blots where you have antibodies that detect maybe some other protein. And so we have to be sure what the exact protein is we're trying to quantify. And so and we should know that based on the molecular weight. So we can look up antibody manufacturer on the website. They'll have a picture of what the protein should look like on a blot and next to like a molecular weight marker. And so one thing we want to do is we can merge the two. So we open the file, select the file that we want to merge. So we want to merge this chemiluminescent signal up in the upper right. I'm going to go to image tools, then I'm going to merge and pick which file to merge it with, which is the white light image. And now we can see that it's been merged there. And so basically now we can say, for example, these bands down here are pretty close to this marker. And we'll talk more about how we want to label some of this in just a second. One thing we could do, you could play around with the contrast. I typically just do auto scale. So that will sometimes bring out some of the background or decrease the white. Maybe didn't do as much as I thought it would be. You could play around minimally with that if you want. I'm going to zoom in a bit. So again, this is a blot for XPA, and we have several different cell lines. The first lane is sort of wild-type Hackett cells. The other 12 are different clones of Hackett cells in which our cells were transfected with a CRISPR guide RNA set of plasmids to then disrupt the XPA gene and disrupt the protein. And so one thing, again, as I mentioned a minute ago, which band is XPA? Well, one is you have to know what the molecular weight of XPA is. So it's about 35 to 40 kilodaltons. This marker over here is 40 kilodaltons, and you could take a look at the membrane or the product catalog for that molecular weight marker series and know which band is which size. And so And then each of these, so this is XPA down here. These are other bands are just simply cross-reacting bands that show up, that react with some other protein in the cell. And so one thing we want to do, or a good habit to get into, is always labeling your blots. So, you know what samples you ran on the gel, but there's no labeling in the actual data file that gets generated. So what we're going to do is do that. See, there's some annotation tools over on the left here. So annotation tools, we can put some text. We can say wild type. We can change the font size if we want, make it a little bit smaller. Then what we're going to do is label all the other ones. So all these other ones are essentially different clones of cells that were grown out following transfection. Look at the spacing here in a minute. So that's there, a little bit higher. So again, it's always a good get in the habit of labeling everything. Sometimes it can be advantageous to label the original data file. If you look at manuscripts that have Western blots, often they only show a very small region of the blot, maybe only the bands of interest. And that's fine. It saves space in a manuscript file where you may have let's say you probe for 10 different proteins. That takes up a lot of space. Each of the individual blots is this size. However, one of the things that reviewers may want to see in some journals require is the original Western blots. Like the full blot, nothing cropped out. That way the reviewers and the readers can see everything. And nothing is being manipulated. Okay. Another thing we might want to do is label some of the markers. This size a little smaller, maybe that small. Again, the advantage is just allows the reader to know, okay, this really is XPA. We know XPA is a certain size, and then they could judge for themselves that you know what you're doing, right? It really is the protein you're saying it is. Okay, and you can do the other markers. This is 170, 130, 170, 55, 40, 35, and 25. Again, all that's based on the product catalog, and which is posted by the gel running station. And we could also, if we want, maybe label what we probe for. Let me make this a little bigger here. So we're going to say that's XPA. The other thing we could do is crop it. So again, with a lot of empty space we don't need. So if you go under image tools here, I guess I don't want to crop it now. You lose that information. You should have cropped it first. So and you can also crop it in other software. So what I'll do is at least export this as a file. So now you have everything labeled and annotated. So you could always go back a year from now, 2 years from 5 years from now, and see what the results of this Western blot was with everything labeled. And chances are you might forget which lane was which clone, years from now. So I'm going to save all this in a folder that was used for this Western blot this day, and maybe I'll label it, say, annotated. Again, the file format, I would encourage you to come up with a file format. I'd like to have the date, what I probe for. This is the exposure length. Uses some information that essentially one wild type was one of the samples was overexposed, and it was merged with the white image for that date. I'm going to save that. I'll save it just as JPEG, make it a little smaller file size. So now we have some data, or something labeled that we can go back to. Now, when we quantify the bands with the densitometry using the software here, I usually do not quantify the merged image where we've merged the chemiluminescent signal with the white image, in part because sometimes there's air bubbles on the white light image. Again, that white image is simply just a white light picture of what the membrane looks like. Sometimes there'll be air bubbles under the blot, and that can interfere with quantitation. So, I mean, this blot looks pretty good, but nonetheless, I'll go back to the original blot, the original chemiluminescence sample, and then what we're going to do is quantify that band of interest. Again, you always want to be sure you know what you're quantifying based on the antibody you're using. And one thing you can do, we're going to draw boxes around this area. We want to make sure things are straight. Sometimes, let's say, you didn't put the blot in there in the machine straight, so now you have something at an angle. Well, that's one that looks bad for presentation purposes. In the software, you can rotate them. So there's left, horizontal, vertical, you can flip things like that. We just want to basically straighten everything out. So basically just kind of hit that custom button and then rotate this little axis thing here too, so that the line goes straight through the band of interest, right click and hit rotate. So everything is straight. I can zoom in up at top or hitting control and zoom in on the mouse. And then what we're going to do is use the image quantitation tools. And there's a couple different ways you could do this. I'm going to use the way I typically do. There will be another link to a YouTube video in the ChemLab handbook that has a really Biorad's, somebody from Biorad showing really a more elaborate and more sophisticated way of quantifying bands. Also perfectly good to use if you want to take a look at that as well. So we're going to go down to this volume tools. So we're going to measure the intensity of that band. And I would note XPA often runs as a doublet, so we see two bands, and it's due to a proline isomerization within the protein. But both of them are the full-length XPA protein. So we're going to draw a box or rectangle around the band of interest. Ideally we use at the same size box for every sample, just because if there's any, just so we're not missing anything. So what I typically do is draw a box around whatever the biggest sample we want. So this one looks like it's the most intense. This one over on the left or on the right here. So I'm going to draw a box, and I'm really going to start on number 1. So I'm going to basically copy and paste this on every sample. Again, some of these lanes don't have a band, or the band looks like it's missing, and that's was really the goal of the experiment, was to generate cells that don't express XPA. So copy and paste all these along for every lane of the membrane. I want to make sure things don't overlap. We don't a box for one lane to be covering one that isn't that sample. A couple more than you need, but we'll just delete them. So we have 13 lanes, the 13 samples were loaded. Okay, so we have a box around every sample. We fixed that one. Then what we do is we come up here to analysis table at the top. Something at the bottom will drop. And again, we're doing a volume table. So it gives you lots of numbers, each sample number based on the U1, 2, 3, 13. There's other labeling that you can do with the software. I don't usually do. What we're interested in is the adjusted volume. This takes into account any background signal differences from lane to lane. That's what we'll use. So what we'll do is up here, we'll export to Excel. Should load up here. File upload or open here. And there's a lot of other numbers we don't need. The only thing we need is this adjusted volume. That's the intensity of the band we're interested in. So I'm going to just select the other columns we don't need and delete them. Like that, everything's centered. This is going to be the XPA band we're looking at. Get rid of some of the extra digits here, and then we'll label it. So this is 1, 2, or wild type packet cells, and different clone numbers. A couple of extra digits there. So that's the expression. The software we're using, again, we're using Azure Imager 600, but using Biorad software to quantify it. So the image, for reasons I'm not 100% clear, it generates essentially a negative value. And we'll take this, basically the bigger it is from 0, the more intense the signal is. So for example, sample 10 here was the most intense, and you can see that over here. This over here is the most intense. So it's the biggest number, biggest negative number. Ultimately, we're going to do the same thing with the Ponceau stain. That'll also generate a negative number. And then we will normalize things. Some of these samples have positive numbers. You can't have a negative intensity. So if it becomes positive, it just means the signal is so low it's less than the average background signal. So basically, we'll just call that a 0. So anything with a positive number, we're going to say 0. And ultimately, looking at the blot, we'll see there's no band there. So it's not, I mean, these are just numbers. The numbers should make sense visually based on what you look at the Western blot. And if they don't make sense, then there's something wrong with your boxes or quantitation. So again, we look like there's some differences in expression, but we want to normalize it to the total protein. So we'll come back to the Ponceau blot and do the exact same thing. So what we're going to do is draw a rectangle around an area of the, switch, I'll have to go back here, click on rectangle again. And so we want a rectangle around some region of the Ponceau plot, this stain. It doesn't really matter which one. I tend to stick with somewhere in the middle of the blot. If you go too high up, there's just less protein there. So usually somewhere where there's reasonable strong signal. And ideally, you could quantify multiple regions of the membrane, and you should still get the same result. Sometimes if things do kind of curve up or down, and you can rotate things, but you draw the boxes as best you can around the same area of the same area bands on the blot between all the samples. Paste. Okay, I think it looks pretty good. The higher low, because you kind of, the idea is it should be covered the same sets of bands. All the bands should be identical. The intensity might be different from lane to lane, but the same bands should be there. So then we're going to do the exact same thing. We're going to hit analysis table. It will give us some numbers here. We'll open it into a new Excel spreadsheet. The only thing we care about again is the adjusted volume here. So we're going to count those extra digits here, center, copy. And we're going to just go back to the previous Excel file and paste it over there. Okay, and so again, we have the intensity of the XBA band, the intensity of the Ponceau, which is the total protein. And again, just looking at the blot, certainly compare lanes 1 to 2, there's a lot less protein in 1 versus 2. And so when we look at the XBA band, we have to take that into account, that although there's less protein between lane 1 and 2, there's no lane, no XBA band in lane 2. It's probably a knockout selling. But nonetheless, we want to be sure that we're normalizing the XBA expression to the total protein. So what we could do is just go back to that file, take the ratio. So we're going to do formula equals that divided by, so E3 divided by F3. Copy it all the way down the, oops, I did that, stretch it out wrong. And we'll get rid of all the extra digits here. And so again, this is sort of an arbitrary number. So what we want to do also is to normalize. And so again, and how you normalize depends on the particular experiment you're doing. So oftentimes we'll make whatever has the highest ratio, highest signal of protein to Ponceau, an arbitrary value of 100. In this case, what I'm going to do is say the wild type, that's our 100. And so that's our normal protein expression. Everything else we're going to compare to that. So what we're going to do is make a formula where we divide the sample ratio by itself. So G3 divided by G3, typically multiplied by 100 to get a sort of round number. We want to normalize everything to that. So if you put the dollar sign between or before both the column and the row number, you can then, it will keep that constant. So if we copy, paste that down, I get rid of all the extra nonsense digits. Now, if you look down here, so you go down, so again, G3 divided by G3, now G4 divided by G3, G5. So again, it normalizes each of these signals, each of these numbers to the wild type. Now let's graph the data. So we'll select, basically scroll down there and hit Control and select the other set of data. So the wild type and cell number will be the cell line number will be the x-axis. This will be the y. And then we're going to insert just a regular graph. Because of the numbers, it sometimes we'll put it both in there. So we can go or both think it's two types of numbers you're trying to graph. So we're going to select data. We can get rid of which one's which. That's fine. Yeah. So you can always edit this. So this is the second series. The edit. We don't really. Yeah. So that is the. Sorry. Go back here. Oops. What did I do here? Okay, horizontal axis labels are it's here. Okay, and then the y-axis are these numbers here. So you can always change that, and it can be useful to double check sometimes. You never want a graph without x and y-axis labeled, or else how do people know what you're trying to show them? So again, the x-axis is the cell line. So we go under add chart elements. You can do x title, x or y. Vertical is, let's just say relative XPA protein. And that, again, it's not totally descriptive because what relative to what? So in a figure legend, you'd say all of the samples are normalized to the wild type cell line, for example. Some things I don't like about Excel is it makes the font size kind of small. So I usually like to increase the font size to 12 for everything, except the axis I like to make bold, a little bit bigger. Just makes it pop out a little better. And get rid of the series name, get rid of the chart title. Not too important here, in my opinion. So now we have a graph here, a graph that has that data. Okay. And so one thing we could do, we've got that data. Oftentimes it can be useful to put everything together. So the file we had, picture we took, we can also insert picture. So I'll go find that folder. Oops. I think I inserted it wrong. So we want to insert picture, place over cells. I think I inserted it into the graph. Oops. Should be able to. Yeah, we can crop this in Excel too. Get rid of all that extra stuff there. If you want, you can try to have a line up under the graph, so like people could look at the protein lane and the quantitation very easily, just going up and down. It can be useful to put the PowerPoint in, or the Ponzo in there. So let's go back to the Ponzo stain. We can export that. It will say it's a low resolution, just the white light image is not quite as strong or as high resolution as the chemiluminescent. Not a problem. We'll export it as a, you do TIFF or JPEG. Do JPEG for now. Okay, so we can go back to the Excel file. We want, we can insert that. Let me find this Ponzo stain. So again, there's different things you could actually export a picture with the region of the boxes you've shown. I could have done the same thing with XPA actually. And that can be useful. Again, you can always save more files. But this allows you to, again, at one time look at the graph and the original data. And so ideally, numbers don't mean anything on a blot necessarily. They should reflect what the images actually look like, right? So there's about a 4 or 5-fold difference between lane 1 and 3 in terms of the intensity in the wild type and clone number 2. And so you should be able to sort of visually say, okay, yeah, that looks like it could be 4 or 5-fold higher. Now, sometimes you'll see people show data where that doesn't appear to be the case. And that could mean they quantified their data incorrectly or something. So, and people should be able to look at Western blot in your graph and say, yeah, those two things line up just fine. So you can put everything in Excel if you like. What I like to do then is to actually put everything in PowerPoint. So this is just this similar data I previously done where I have, and I like a title that has the date. So I can always go back later on and find that original date. Again, all my files have dates in them, so I can go back to those files and find the original Western blot, description of what I did, Western blot analysis of XPA protein expression and wild type, several XPA knockout clones. We've got the annotated merged image between the chemiluminescent and the white image, got the Ponzo stain in, and then there's some quantitation. So, and this is a good way to present data at maybe a lab meeting or informally. And for a publication in a manuscript, you'll see nobody shows full blots. That would take up a lot of space. So typically what people might do is, I just show around. So even in PowerPoint, you can do this. We can, picture format, let's say crop. So in reality, what people would show in a scientific manuscript is probably just this area, right? Because all you care about is that. That's the protein you're interested in. You're not interested in the cross-reacting bands. So that's often what you'll find in the manuscript. Sometimes in supplementary information, which journals will require, they want the full blot, nothing cropped, because they want the original data. The science is changing a bit in that it's becoming more common that essentially all your primary data needs to be reported. Make them do that. So because it makes it easier for people to see what you actually did and not like you're trying to hide something. Again, that's a way to use ImageLab to quantify Western blot data, export the quantitation, the densitometry into Excel, and then present the data in a PowerPoint slide.